Okay, I'm just going to pick up where that video left off. So we're looking right here at two identical DNA double helixes, one with a old strand and one with a new strand. And so it's going to talk about that right here.
Identical double helix DNA molecules from their one original double helix DNA molecule. We call this semi-conservative replication because the two copies each contain one old original strand and one newly made one. Oh, one last thing. Surely you've had to proofread your work before to catch errors. Well, you definitely don't want DNA polymerase to make an error. If it makes the wrong DNA base, then you could have an incorrectly coded gene, which could ultimately end up in an incorrect protein, or no protein. DNA polymerase is just awesome. It has proofreading ability, which means it rarely makes mistakes. And that's a good thing. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right, so I love that I kind of reviewed through all the enzymes. So we're going to hop over to our notes now. All right, I need you to turn to page 5 in your notes packet, and we're going to take some notes on this page going over DNA replication. Okay. So the very top, you can see DNA structure. It has two primary functions. And the first one, as we could guess, it is to store information to deliver the activities of the cell. So it stores the information in a code, and then it's able to, make your proteins and make your body function. Okay? Now, in order to make new cells, we have to be able to copy our DNA. We got to replicate. So it says copy itself exactly for new cells that are created. Okay? So DNA is a genetic code that tells our body how to function by making proteins, and we need to be able to copy the DNA. If you think about it, if you get in a car wreck or you, fall off your road bike or your pedal bike, whatever, and you fall on the road and you scrape up your body and you kind of got asphalt, turf, scrapes and burns all over yourself, you got to understand those cells get scraped away. They're destroyed. So your body's got to constantly make new cells, okay, to repair the skin tissue that got damaged. Or maybe it's the stomach lining from the acid in your stomach. It's getting broken apart and eroded away, so it's got to continually make new ones. In order for that to happen, you got to copy an original cell into two new cells. And that's why DNA structure is such a big role when it comes to replication. Okay, so here is kind of the whole overall process where we take DNA and we use it to make our proteins. Okay, it is a 3-step process, and we'll get into this next unit. But the part we're going to focus on right now is just the DNA replication process, okay? Where DNA makes more DNA. All right, so when does this happen? We'll just continue on with the notes. Okay, DNA is exactly replicated during the S phase. If you guys remember what this term was, of interphase. I'll pull this up. So it's S phase of interphase. Okay, that's when our DNA is getting replicated. And you know what? It doesn't take very long for a cell to copy all of its DNA. Okay, looking about an hour or less. If it's a human cell. Different species, different animals can have more or less chromosomes than us. Okay? All right, so where does this happen? This is all inside the nucleus. Okay, just like the video told us. How, at many points along the DNA molecule, an enzyme will unzip, okay, the two strands, breaking the, what bonds? The hydrogen bonds between the base pairs. So how does it do it? By getting unzipped, broken apart, and then it gets ready to replicate. All right, so here is a DNA double helix. Here's the original, right? You have the two strands, and look at these base pairing rules, A with T and C with G. 2 hydrogen bonds, 3 hydrogen bonds. Now, you don't see these other molecules, but you know this is sugars and phosphates, right? So they're all twisted together, and then, bam, it got broken apart right here. Okay? Now, there's 3 enzymes I want you to know. And the first enzyme here, that was the helicase, and that's what breaks apart the hydrogen bonds, right? And then the other enzyme is what does this right here. It's creating these new strands. And that was the DNA polymerase. And I always think of polymerase as Polly the angel, and angels fix things and makes things better. Okay? Where the helicase, it's little hellion, and it breaks things apart. Okay? And then you have the ligase. And the ligase is what fuses the new strand with the old one. Okay. All right. So there's a key term. I want you to add this to your picture that you have on page 5. I'm going to flip this camera, see if we can switch the view. Oh, we'll just come right here. So, how well can you see that? Oh, that's bad. Oh, this way. Which way do I go? It's down here. This is entertaining. All right, that picture right there. I want you to label and identify where the replication fork is. That was a lot of work. I'll figure out this camera thing. All right, so draw a little arrow, and it's the point where the DNA double helix, it's broken apart, where it can do replication. So this is what we call that fork, right? It splits into 2. Okay, now, this is something I do want you to add to your notes. You have some space there underneath number 3. And so we're going to add this. And granted, you don't have to add it word for word. But let's just take a look at it. All right, the bases of each exposed strand will attract the complementary bases floating freely in the nucleus. So once you have that DNA double helix and it gets broken apart by the helicase, now we're going to add the complementary bases that are just floating in the cytoplasm. All right. So maybe in your notes you could put something in the effect of complementary bases are added to the strand. All right, next one. The result is 2 new DNA molecules, each of which consists of an old strand and 1 new strand. So really, if you draw, you already have this picture, if you even just labeled new strand, old, new strand, old. So just to remind you that we've got a new with an old. Okay, so complementary bases are going to be added, and at the end, you have an old strand with a new strand that DNA polymerase helped build and create. All right, so here's our old, and there's your new. Oh, sorry, that was really fast. Old and new. Okay. All right, so we'll actually move on to this next part. So here's what I want you to put in that box, that table you have there. It says the enzyme jobs. Let's go ahead and get to the helicase. And the helicase job is really to unzip your DNA. All right. So if you just put unzips DNA, you're going to have the basic idea of what it does. So the helicase unzips DNA. It breaks apart those hydrogen bonds. So we look here. Tells you that means it breaks the hydrogen bonds to hold the A's, T's, C's together. So here is your DNA double helix. Once again, hydrogen bonds are holding this together. And the helicase is going to start right here and break it apart and create the replication fork. So here they're hooked together, and here they are separated. So that is your helicase. Now, this picture's kind of funny, or this cartoon's funny. So here we are. Here it comes in. So you have a nitrogenous base here, and a nitrogenous base here, and bam. Here it comes and breaks apart the hydrogen bonds. Right? So this is an example of water molecules that have been broken apart by a hydrogen bonds. All right, moving on. Okay, so the next one, that DNA polymerase. Okay, it's going to add new nucleotides. All right, so once again, here's the DNA double helix. The helicase broken apart, so here's the replication fork. And if you remember from the video, these are the primers that the primase brought. And now the polymerase knows where to start. And then it's going to start adding nucleotides according to the base pairing rules. So you can tell what direction this is supposed to go. Remember, the RNA polymerase is always going to go from a 5 to 3 direction. So it's going to go this way, where this one's going to go that way. So they go in opposite directions when you're building it. Okay? So there's your DNA polymerase. And you could put that it adds nucleotides, creating new strands. All right, last one is the ligase. And if you remember about the ligase, your ligase is what glues the old with a new strand. Okay, so glues new nucleotides to the strand. So we're looking here. The black is the old parent strand. Here is the new strand. Okay. and it's this ligase that's putting the old with the new, creating new hydrogen bonds. All right, so here is our, I love this picture because you kind of see it from the chromosome view. And remember how that chromosome's all coiled up and coiled up and coiled up. Well, if we uncoil it and uncoil it, then you get to your DNA double helix. So here's those nitrogenous bases, the sugar phosphate backbone, and bam, here's the helicase enzyme that's breaking it apart. There's your replication fork. Come over here. There's the polymerase that's adding nucleotides according to the base pairing rules. And then here is another polymerase that adds nucleotides. So what is the enzyme that's not mentioned, or you don't see it on this? And that is your ligase that actually fuses these together. So here's the old strand, and there's your new strand. Okay, old strand, here's the new strand. You don't need to worry about the terms leading and lagging. Kind of like I mentioned earlier, they got cut off. Okay. You can skip all this. All right, so here's what I'd like you to do. Just anywhere on your notes packet. You can do anywhere on this page, maybe towards the bottom, the top. But I want you to draw a zipper, all right? And I want you to label the parts here. So here is your little zipper. You can probably get an idea of already what's happening here, right? Here's our DNA double helix. And this helicase is going to break apart those hydrogen bonds and unzip it. So I want us, let's label these parts here. So this would be the helicase. Okay. And now we have in a strand, okay, two strands that are the nucleotide bases are just exposed. So now we need to add new nucleotides according to the base pairing rules. So if this nucleotide right here, okay, one of the teeth on the zipper, so one of those, that's a letter A, then we know this one over here is the complementary base, so that would be T. And if this one's C, we know this one's got to be G. Okay. So now let's talk about those bonds, right? This zipper can go up and down as many times as you want, nice and easy, because these hydrogen bonds are really weak, easy to break apart. So hydrogen bonds are between the teeth of the zipper. And then this strong fabric where it's all sewn together, these are those strong covalent bonds. Okay. So what I wanted you to label on your zipper is hydrogen bonds, covalent bonds, and then I want you to put the helicase enzyme. And then what else could we add here? What would be the enzyme that would be working? And we'd have an enzyme here, and we'd have an enzyme here. One enzyme's going this direction. This enzyme's going this direction. Okay. We know this is the replication fork, which is where our helicase is. So these guys right here is your DNA polymerase, right, which adds nucleotides. I'm just going to put poly. Alright, so they're DNA polymerase. So you could compare the zipper to the same thing that's happening over here. Here is where I'm going to put an H. There's the helicase. Okay, and then you would have a polymerase here, you'd have a polymerase here, and they are working opposite directions. And then you, of course, have your ligase that's going to glue the new strand to the old strand. All right, so you got that drawn somewhere. You probably do it at the very bottom, hopefully. You found a spot for that. All right, pause this really quick. All right, so here I kind of just added more of the information I'd like you to draw with that zipper. But like we said, covalent and hydrogen. This is your sugars and your phosphates, and here's those bases. And we know we've got our replication forks. All right. Like we said, I got our sugars and phosphates, A's, T's, C's, and G's in there. All right, so at this point, that kind of finishes up DNA replication. And if you have any other questions, just make sure you look through, and on Canvas, I do have some videos that I skipped over on the PowerPoint that you could watch that will help you. All right, that's it.